take you guys initially. Okay, I like to initially take you guys through something that would be a bit more common to you guys, something called lung adenocarcinoma. So, lung adenocarcinoma is the most common type of lung cancer, and it's very prevalent in women and non-smokers, especially. And also in non-smokers, the major cause of this particular cancer or incidence of this particular cancer is genetic mutations and mutations like EGFR and ALK. Now, EGFR and ALK are two proteins within your body. Now, along with that, we'll focus on one particular protein. Here, we're taking something called ALK. Now, what happens in this particular scenario is there are these mutations that lead to this breakdown of this ALK gene, and that ALK gene gets fused to another protein called or another gene called EML4. So this creates like a fusion protein where the N terminal or the starting point of the protein is that of EML4 and the C terminal or the bottom part of the protein is that of the ALK. So once this fusion protein comes out, this is what happens. It leads to constitutive ALK kinase activation. Now, as you all may know, in our body, everything works through different pathways. So every epigenetics that you see coming out of your, every trait that you see within our body is a result of certain pathways happening. So, like that, we have a few pathways like PI3K, MAPK, JAK-STAT pathway. All of these pathways are involved in cell proliferation, cell division, cell growth, and so on. Now, ALK is one of the protein within that pathway. And when this fusion protein between EML4 and ALK, when these fusion proteins are generated, it leads to ALK being activated like all the time. So it's constitutively activated. And when it's constitutively activated, it's constitutively giving a downstream signal to pathways such as MAPK or JAK-STAT pathway to continuously generate or proliferate your cells. And there comes or that leads to what we see as lung adenocarcinoma. And this particular arrangement that we see is present in around 3 to 7% of the lung adenocarcinomas that we see. Keeping in mind that it is the most common type of lung cancer out there, 3 to 7% is a pretty good number globally. Now, how do we deal with this particular ALK mutation, or how do we stop this? So, again, you use drugs, right? You have something like all of these drugs are drugs that were approved for cancers where ALK is involved. So, you have your crisatinib, which was approved in 2011. Then you have your ceritinib, which was approved in 2013. Another one which is approved in 2015. 2017, another one in 2017. So you see that there is a bunch of these drugs that were approved for ALK, kind of showing you how much of an importance that ALK-based mutation and ALK-driven cancers play a role in the progression of that particular disease. So till here, it might be pretty familiar. You have a particular a tumor type, a cancer type, and you take a particular drug for it. But how does these particular drugs, how do they act within your body? So these drugs, what they basically do is they kind of bind to the protein of interest or to your target of interest. For example, this is an image of a protein. So I'm not sure how many of you are familiar with how a protein would look like. So you have your alpha loops and your beta sheets. And this right here is the crisatinib, which is the first drug molecules that was approved for ALK. So this is a 3D image of how the crisatinib is in action. It goes and it binds to your ALK protein at a particular site. Now, what will that lead to? So it leads to something called inhibition. So these are the two main topics that I would like to talk about, and I would like to show how it's being designed within our platform. So the two main topics is inhibition and binding. So for an inhibitor or for a drug to act within your body, it needs to perform
Aman,
inhibition. I'm sorry, was there a
I think it's background noise. You can continue.
yeah, sure. So for a drug to act within your body, it needs to perform both binding and inhibition. In this case, because it's ALK protein, we're talking about inhibitors. Because what we want to do is we want to inhibit ALK, and we want to stop all the downstream pathways from leading to, or downstream pathways that leads to cell progression or cell proliferation. So in this case is inhibition. So for particular ALK, and if you are designing a new drug for ALK, we want that particular drug to both inhibit and bind. And what I mean by that is, or what I actually mean by this particular writing is that for inhibition equal to binding, what I meant is inhibition. For inhibition, we require binding. So inhibition requires binding, but binding does not always lead to inhibition. So for a particular, say, uh, in this case, your crisatinib, it has bound to the ALK protein. So you have your protein right here, and crisatinib is bound within it, and it is inhibiting ALK. But not all the binding, not all binding of molecules to proteins can lead to inhibition. So what are all the factors that you need to keep in mind when you are inhibiting a particular protein? So some of these factors I'll introduce. So one of them is that we need to ensure that this particular molecule or your drug molecule or your possible drug molecule, candidate molecule, is bound within the pocket and bound within pocket in such a manner that it will bind to certain residues which we call as key residues. Now these residues are residues, so as you know, proteins are made up of amino acids, right? So though the protein is made up of multiple amino acids, for the function of that particular protein there are a few amino acids that are found to be more relevant, or the basically not for the structure but for the function of that particular protein. So what this particular crystalline or any drug molecule does is they bind to a pocket within that ALK protein which is involved in ATP, which is ATP kinase for phosphorylation. So in the same pocket, this particular drug molecule also binds. So we need to kind of bind to these residues which are involved in binding of the ATP as well. So it's not just that the molecule needs to bind to that particular pocket, it needs to bind to the residues that are called the key residues. Then that binding should be high affinity binding, that the binding should be strong enough. So these interaction with these residues will cause, they are like the glue. So the interaction between the atoms of your drug molecule and the atoms of the residues within the binding pocket are the ones that glues the molecule into that pocket, and we need to ensure that this gluing will give a high binding affinity, like strong affinity within the pocket. And we also need to ensure that these affinities are stable. So as you know, your protein will not be always static within your body. It might be in the cytoplasm, it might be moving, or even if it's a membrane protein, it will have constant movement. So even during the movement, we need to see how stable your particular protein is within that binding pocket. So, how do we assess all that? How do we say whether the molecule is stable? How do we say whether the binding affinity is good? And how can we generally evaluate whether a molecule is a potential strong binder? And only if it's a strong binder can it inhibit or activate your particular protein. So, in lab, you have multiple different pathways. You have assays, you have your cell-free assays, you have your cell-based assays. But how do you do it in silico? So, for that, I'll show it through our platform. We do something called molecular docking. So before I get into molecular docking, this is your Boltzmann platform. On the side, you have a lot of these options. Along with that, you have your modules. So within Boltzmann, you have your protein engineering modules, which for which the session will be taken right after this. You have your small molecule, you have your synthetic chemistry, and you have your RNA design. So all of these can be or tools for developing and designing and evaluating and validating all of these tools will be provided within our platform. So I'll go directly into small molecule design. So you have a bunch of these options over here. I will not go through all of it. I'll go through two of them specifically. So we'll go into something that studies binding. So that particular thing right now is called molecular docking. So what molecular docking does is it will take your protein structure. So before I show you guys something, there is something called PDB. Okay, PDB is a database that stores three-dimensional structures of your protein. So any proteins within your body, they will have multiple methods, NMR or X-ray diffraction, different methods wherein which you can get the structure of the proteins. Once you get the structure of the particular protein, we can use the structure to kind of lead on how a particular molecule will bind. For example, right now I'll just give some name, and what I'm going to do is I'm going to just give molecules. So there is different ways you can represent small molecules, but the most easiest way to represent a small molecule is this one dimensional form called smiles. So using the smiles you can represent the molecules, and once you have these, um, you can just give me you can just take them and then you can upload them as just the smiles. So for example, I've uploaded the file, the same file that I just showed you. And I'm just going to take one of the PDBs that I found in that database, that three-dimensional structure database, and I'm going to add the protein pockets. So mm, just a minute. So that doesn't confuse you more than it already does. So this is how your protein looks. This is a three-dimensional structure of the protein. And then this is what I was talking about when I mentioned your molecule. So now you can see that you see one that is yellow in color, that is your crisatinib. And then you see something that is in a cyan color, that is the ADP molecule. So in the same pocket that an ADP should bind, and only once an ATP gets converted to ADP will that molecule gets activated. So to prevent that activation, what happens is this particular crisatinib, it will go and bind to the same pocket. That is called competitive inhibition. It will bind to the same pocket and cover the pocket, and it will bind more strongly than ATP, that even if the ATP comes to that particular pocket, it cannot dislocate it. So this particular binding within this particular protein is what causes the inhibition of the entire pathway, and that is what causes, or that is why we use it as a treatment against different types of cancers. So what we are trying to do with docking is basically we are trying to replicate the same but using in silico methods. So for that is why we are going to perform this particular docking. So now you remember the binding pocket that I just showed you. So you can just using, once we have understanding of that particular pocket, you can just take out you will get these values. Now these values will be defining within the protein structure you gave, where all exactly can you find the pocket. So the protein structure is pretty huge. Where does it needs to kind of put that molecule to prevent that, prevent the binding. So that is called the ATP binding pocket. So you can just put the centers and the diameter or the size of that sphere, and then you can just run it. So because it might take a bit more time than what is required, I'll just show you how the output looks like. So this is your output. So it would give you, say, these are the different poses. So I gave around, say, in this particular experiment, I gave around 484 molecules. So for all the 484 molecules, what it did is it tried to find the binding pocket. So this is your protein. Within the protein, how does the molecule bind? So this is your binding conformation. And then if you remember, I showed you this and I said you have to identify the key interactions. So to identify the key interactions, you have an image over here. This will show you which all residues is it interacting with, what is the distance of the interaction, what are the type of interaction. Now this is a bit more of chemistry, it has a bit more of chemistry part into it, but it shows you certain interactions are more stronger than the other, so it covers that part as well. And then you have binding affinity. So for that, binding affinity is why you have something called the score. So the more negative the score, the higher the binding affinity is. So the score is like what we call as free energy of binding. The more negative the score, the better the binding. And then you have something which is the stability. Now for the stability, you would have to do something called molecular dynamic simulation, which I'm not getting into because it has more of a physics base to it. But basically, you will be trying to move the protein around and see how strong the binding is. So that is how you basically analyze how well the protein is binding and where all the protein is binding and what all residues it binds to. So only if the molecule is a good binder can it inhibit, like I mentioned here. Only if it binds can it inhibit. But not all binding will lead to inhibition. So what does that particularly mean is that, one second. So in this particular protein pocket, you can see that this is your protein, but then you can see that there is multiple crease and crevices over here. The multiple pockets that can be formed. Which pocket do you want to choose? So for that particular reason, you have to predict the uh, inhibition. For predicting the inhibition, because there's no fast and quick method to predict inhibition, what we do is we do something called QSAR. So if you have the molecule and you know the activity of the molecule or what is its IC50 value. So if I'm not wrong, most of you know what is inhibitory coefficient 50. It is the 50% the concentration where in which the molecule inhibits 50 percentage or sorry, my bad, the half of the 50 percentage of the concentration that like for example over here you have your cells. So the concentration at which 50 percentage of the cells are inhibited, or in this case we are trying to kill the cancer cells. So the concentration at which at least 50 percentage of the cancer cells are dead, that is your IC50 value. So if you have any value, not just IC50, you have IC25, any value that is associated with inhibition, you can build a QSA model. And the QSA model does nothing, but it's a quantitative structure activity relationship model. So to break it down, quantitative means an ML model. Structure is the structure of the molecule, and activity is the IC50. So it is an ML model that bridges the structure of the molecule with the IC50 of the molecule. So if you have built a QSA model, what you can do is you will build the QSAM models with molecules whose activity you don't know. Sorry, and then you use the model to predict what is the activity of a molecule for whose activity, sorry, activity of a molecule that for which the property you don't know what the value is. For example, I'll show you some module called property prediction. So in this case here, it is it is library. I'll choose any library. These are nothing but molecules and their smiles, like like I showed for docking. So I'm going to choose a small set of molecules. And then as you can see here, you have their smiles is smaller, but then you have your smiles here, and then I can choose any model I want. So within the Boltzmann platform, we have a set of models that we have already built. Now these properties are pharmacokinetic properties like half-life and bioavailability, some toxicity parameters like carcinogenicity and so on. You can choose any of these properties you want. These properties, all of these properties are all QSA model that we have built. So using that property, I'm going to predict the values for molecules from this particular library. And for these molecules, I don't know what their values are. I don't know what their bioavailability values, their hydration free energy, or their lipophilicity. So I'm going to predict it. If you have built any models of your own, you can choose that as well. And then you can just click submit over here. So I will just pull up some activity. So, so this is something I ran before, not exactly for all the properties that I've selected right now. So over here, these are two different properties I've selected. BBB stands for blood-brain barrier permeability. So it's a classification model. If a molecule is permeable, it will say 1. If it's not permeable, it will say 0. So in this case, it predicted these two molecules as 1. This is another ABC receptor protein, which is a protein that is dealing in cancer, so for drug resistance against cancer. If a molecule is found to be an inhibitor of that particular protein, which is called PGP, P glycoprotein, it will be showing as 1. And then I have built some other models. So these are some of the models that I have built, and you can predict the activity again, how strong it is, or the IC50 value, again using them, using these, you choosing these models in the property prediction and the custom property models as well. So these are just for predicting. If you want to build the model, you can go to custom property model. I'm not covering that module right now. But if you just give, like as it's mentioned in the slide, if you have a set of molecules and you know their activity, and if you give them as input to the library right here, you have library right here, you can build a particular model, and these models can be used to predict the activity of any of the molecules that you want to predict the activity for, but you do not know the values for. So that is basically how, the Boltzmann platform helps you not just in predicting or studying the inhibition of the molecule, but also for studying the binding of the molecule. And both of these can be run in sequentially. You can run inhibition prediction followed by docking, or docking followed by inhibition, so as to make sure that your molecule is not just binding to the protein, but is also inhibiting the protein, and and is a possible drug molecule out there. So that's all from my end. Is there is there any doubts, or we can move on to protein engineering?
Participants can put their doubts in the chat box. I think they may not be having any doubts, so you can go forward with the protein engineering module.
Yes, Pawan, thank you. Hello everyone. I'm Harshvardhan from Boltzmann Labs. Today, coming to the protein engineering, we have in protein engineering, we are also having separate tools for protein engineering and then enzyme engineering. So I'll just show you around. So today I'll be talking about how AI is transforming enzyme engineering and speeding up life science research. Traditionally, enzyme engineering has been slow, experimental, and expensive, but with the modern AI models that we have now, we'll be able to predict, design, and also optimize the enzymes much faster than ever before. And so basically, what is enzyme engineering? So enzyme engineering essentially means modifying enzymes so that their performance or the functions will be much better, and they'll be faster, more stable, and also more specific. We'll be using three major strategies directed evolution, design, and AI-driven design. So basically, directed evolution is where we introduce random mutations and select the best performers. And coming to the rational design, we'll be using structural knowledge to make targeted changes. In the AI-driven design, it helps us to predict the most promising mutations before we even begin experiments. So together, these approaches allow us to push the enzymes that are towards the industrial, medical, and research applications. The challenge is that the mutation space is unbelievably huge because there are millions of possibilities for even a single enzyme. Testing them experimentally is very slow and very expensive. And that is where AI comes to the picture. AI will help us to reduce this burden by learning from existing biological data and also predicting which mutations are worth testing. This means we can escape like almost 70 to 80% of the trial and error, and also focus on the most promising candidates. So how AI is helping in enzyme engineering? AI models will be looking at millions of natural protein sequences that are available in various resources, various databases that are available online. And AI will learn what makes these enzymes stable, soluble, or active. With those learnings, AI can predict how stable a mutation will be, and it also be able to suggest modifications to improve enzyme solubility, and also will identify mutations that boost catalytic activity and drastically reduce the screening by 10 to 100 times in some cases. So AI is not replacing biologists, but it is giving them superpowers on how we can be able to engineer the enzymes. And coming to how AI will understand the proteins is just like the language models learn grammar and vocabulary. Protein language models will learn the grammar of amino acids. They'll be identifying the patterns that nature has used for billions of years, like conserved motifs, structural signals, active site residues. So with this internal understanding, the model can predict which mutations will stabilize the fold, which residues from the pocket, and also how the enzyme behaves under different conditions. So coming to the protein language models, these protein language models like ESM, PROT, T5, MSA, Transformer, ProGen, these treats the proteins like the sequences. Each sequence is encoded into a high dimensional embedding that represents the structure, function, and also the evolutionary behavior. So these embeddings are basically the extremely very powerful. They'll have the ability to capture the information about the secondary structures, the solubility, and also the functional sites. So we'll be using them as the foundation for the downstream prediction models. So now I'll take you through the suite of Boltzmann AI discovery suite. And coming to this protein engineering model, if we go to the protein generation, we have enzyme generation tool, again, which has function-based generation and enzyme generation. I'll show you, take you the function-based generation, enzyme generation. So if you give the input as in EC number, like basically enzymes will be having an EC number. So for example, we'll be taking hydrolase enzyme EC. And if we give, and also you can give how many number of sequences you want it to generate. For suppose, say, let's say 10. And if you submit, okay, you can give the experiment name too. So the experiment is submitted, and since it takes some time, I'll be showing you the previous ones. So basically, like this model was trained on the existing enzymes sequence database. Like from this model, like we'll be generating enzyme sequences that are based on the user prompt, which should be a EC number. So based on these sequences, like we'll be able to further work on it. And then coming to property prediction, we have enzyme profiler. You go to the enzyme part. Like here, you can give the experiment name and you can give the enzyme sequence directly, or also be able to upload the file of enzyme sequence and the substrate smiles. Like you'll be having the option to upload a CSV file, or you can build a library and also give that a set of library of enzyme sequences. Like of the properties that you want to learn about that particular enzyme sequences, you'll have the option to select the pH prediction, enzyme solubility, function prediction, and substrate prediction. So this is so here we have this. We have given input, and this is how the output looks. Like you'll be able to see the smiles file. So this is the pH prediction, and the enzyme solubility is 1. And the function prediction, like we have got a function ID, and then the substrate prediction. And coming to the optimization of enzyme, we have this tool called MLDE, which is called as machine learning directed evolution. Here you'll have to give the residues of the enzyme, and and this uses the machine learning guided models, like which will be able to intelligently mutate the protein variants like around the specific residue positions. Like after giving the residues, only those will be mutated, and it will be creating a focused mutant library and evaluates each sequence physicochemical properties. So I'll show you an example like how like we'll be able to get the output. So this is how we'll be getting, like we have different samples, like different sequences. It will give you the length of the sequence and also the molecular weight. And pi is basically the isoelectric point, the hydrophobicity, acidic count, basic count. So basically, like these helps you to learn more about the enzyme sequence. And these are the ACD, like these are the amino acids like alanine, cysteine. So it will give you the probability of that alanine amino acid, the probability of that particular acid being able to present in the generated. So here the probability of alanine is 0.116 in the first sequence that is generated. And also we have the toolbox, which has multiple tools that are related to protein and also enzyme. So if we go to the soluble, ligand and PNN, like it is a structure-based peptide design tool. Like will help you to generate the ligand compatible sequences using the 3D geometry of a protein. Like you will have to upload the protein backbone, the file that is consisting of protein backbone, and also the ligand remaining structure of the protein. Like here you won't be having the complete, like in some cases you will not be able to find the proteins that have complete structure. You will just complex. So you will have to upload that complex file, and you have to give the number of sequences to be generated. And if you click submit, then you'll be getting the output as shown. So here this T is called temperature, and like the seeding number, like the number of residues. And this is the sequence that is generated. So this is how we'll be able to learn about enzyme engineering using different tools. And now Lakshmi will be showing you some more tools in the protein engineering module.
Hello all. Am I audible? Yeah.
Yes.
Is my screen visible? What do I?
Yes.
Yeah. Hi all, this is Lakshmi. I work as a researcher at Boltzmann Labs in the domain protein engineering, specifically antibodies. So today I'll be gonna talk about antibodies. So firstly, before getting into the platform and details of that, I would like to talk about the conventional or traditional antibody designing method. So the conventional antibody designing relies heavily on experimental methods, as you all know, like animal immunization, hybridoma technology, phage display, and yeast surface display, and so on. So these approaches involve generating large number of libraries of antibody candidates and physically screening each candidate against a particular target of choice, then optimizing those candidates through labor intensive processes. While these methods have been successful in wet labs, they are often time consuming, costly, and limited uh, in terms of the diversity of sequences it can generate, right? So this whole thing makes it challenging to rapidly identify high affinity, stable, and therapeutically viable antibodies in wet labs. That is where AI comes into action. So, AI integrated antibody design revolutionizes this conventional drug discovery process by leveraging machine learning and deep learning technologies to analyze vast amount of antibody sequences, structure data, and all. So, AI tools can generate even novel or antibody sequences from scratch, predict different CDR variants of a single antibody, and assess the biophysical properties and model three-dimensional structures of these antibodies and their complexes with antigen and other molecules. So, by simulating interactions and predicting stability, solubility, binding affinity, and several other biophysical properties in silico, AI drastically reduced experimental overload, accelerating drug discovery and enabling data driven antibody design. So, our platform has got three modules, mainly protein engineering, small molecule, and synthetic chemistry. So I would be explaining more about protein engineering. Okay. So how do antibody drug design work? Here I have mentioned a simple workflow. This is just a basic workflow where the initial step is antigen identification. So prior to any antibody identification, the initial step involves identifying the disease target, that is an antigen, mapping relevant epitopes on that antigen. So once you have a disease target like dengue virus or any other virus, the first thing you do is mapping the epitope of that virus from the literature reviews. Then comes the creation of or generation of antibody library. So this would involve the usage of so many tools, AI related tools, which we have in our platform that would create a library of antibodies that can be later on screened against this antigen. So that steps comes here. The second one, which is the library generation step. The third one is the developability screening, which means if you have a set of a library of antibodies, you need to know which antibodies would be therapeutically viable. Right. For that, we have certain property prediction models on our platform, which would help you assess the stability, solubility, and hydrophobicity of the antibody candidates. Thereafter, from a library of 1,000 or 2,000 antibodies, you can just filter out maybe 10 or 20 antibodies, and you can predict the structure of those antibodies later on using certain structure modeling AI tools, which we have in our platform. So once you have the structure of an antibody and antigen, the next thing you want is you have to see if this antibody is getting bound to that antigen properly, to the epitope of that antigen, right? So for that, we need to perform molecular docking using several tools like BOLS, AlphaFold, Multimer, and several other tools which we have in our platform. So this will help you in docking the antibody against that antigen and finding if that antibody antigen complex is stable or not. And other than that, you will have to interact once this complex is formed, you'll have to analyze the interactions of each bonds between antibody and antigen, like hydrogen bonds, salt bridges, hydrophobic contacts, and you'll have to predict the delta G and KD values, which gives the stability of the complex as such. And based on all these criteria, we can really filter out few antibodies which are highly viable, therapeutically viable, and later on we can send that antibodies for MD simulations. MD simulation is more of like creating a dynamic environment where you will find how these antibody antigen complex will get stable in a dynamic way. So that requires more computation and all. So I will not be going through those in detail. Till interaction analysis, I can show you a few tools which we have in our platform. So the first step is, as I told, identifying your target. And once you have your target in hand, the next step is generating antibodies that can fit into that target, right? So either you can go for random antibody generation. That means this tool would generate some random library of antibodies uh, without giving a reference antibody. And the other one is that you can have a reference antibody and you can optimize the sequence of that reference antibody. And uh, maybe if you have an existing antibody in market which is targeting a specific antigen, and you feel like you have to improve the efficacy of that antibody, you can just use tools like CDR generator to really change the CDR regions, optimize those regions, and generate so many variants which are um, different in CDR compared to the reference antibody, and you can generate like as well. So the first one is random antibody generation, which generates random sets of antibodies. You'll just enter the number of sequences you want and an experiment name. This will give you random set of antibodies with heavy and light chains. Both heavy and light chains. Okay. And this table shows the length of each antibody sequence and the molecular weight, isoelectric point, hydrophobicity. All these values would help you in filtering out the best antibodies. All right. So this is just a random library generation method. The other one which I talked about is the CDR generator method, wherein you will give a reference antibody which is known to bind to that antigen, and you are optimizing that antibody for increasing the efficacy of that antibody. So here you will enter the reference sequence in FASTA format, and how many number of sequence you want, you can enter that many numbers, and this will generate you a it will generate the sequences of the antibodies which are what do we say, like the sequences would be same as the reference antibody except the CDR part. It will only change the CDR part, rest of the framework is unchanged here. So likewise, we have several other antibody generation tools like backbone generation, where this tool will generate the structural framework of a antibody, and later on you can generate the sequences from that framework. It would look like this. It will give the structural framework, and you can feed the structure to another tool called antibody sequence to structure to sequence generation tool, which would give the sequence of this generated backbone structure. So now that we have lots of sequences in hand, either from random antibody tool or from CDR generator or from antibody to sequence generator, we have lots of library of antibodies in hand, right? So now the next thing is, we need to understand the properties of these libraries so that we can filter out the best ones from this. So in this property prediction model, you have several features like thermal stability, solubility, CDR prediction, and antibody antigen binding. So here in the property prediction model, it predicts features like this. And once you generate the sequences, either from random or CDR generator, you just feed this into this tool, either in the form of a library, or else if you want to assess the biophysical properties of just single antibody, you can just enter the heavy chain and light chain sequence here. Okay. And you can select whatever features you want and submit the job. Okay. This will give you results in a form like this, which will also give the CDRs of that particular antibody. So if you have 100 um, library of 100 molecules, you can just feed that library into this property prediction tool. This would help you in finalizing the best 10 antibody sequences. So once you have the best 10 antibody sequences in hand, the next is the structure prediction. You have to find the structure of that antibody. For that, we have n number of tools. One is protein structure prediction, wherein you will give the sequence of that antibody, and the tool will give you the structure of the protein. You'll just have to enter the antibody sequence, and this will give you the structure of that whole antibody. It would look like this, and it would also generate some metrics like PLDDT and PTM. PLDDT shows the entire accuracy of the predicted structure. If it is greater than 80, it's highly accurate structure. And PTM is the interface. Yeah. PTM is the accuracy of each amino acid occurring in that structure. That's also great. So in that way, you can generate the structures of antibody, and you can dock it against your antigen structure. For that, we have several docking tools like Megadoc and Bowls too. In Megadoc, you can enter the docking, I mean, the antibody and antigen complexes PDB files, and a doc, it would produce 10 different docking complexes in different poses. And you can filter out, later on, you can filter out from those using the another tools like Prodigy, which gives you the binding affinity values. And we have another important tool, which is Bowls to, yeah. This will also help you in generating the antibody antigen complex, and we can easily find the stability of that complex by using tools like Prodigy. So Prodigy is a tool that helps you in finding the overall stability of that complex. What you need is the PDB file of that complex. You just have to upload it, add the chain, interacting chain. If the antibody is A and antigen is B, you'll have to add it like A comma B, and it will help you in generating a, okay, it will generate a CSV where you will see the binding affinity and the interacting residues. So if you have the interacting residues in hand, you can just check if it is interacting properly or not. And we have several other stability checking tools like RC plot and all, where you will upload the protein PDB file, and you can see if it is stable or not. Yeah. So this is just an overview of our platform and how it works. So that's all from my end. If you have any doubt, you can ask me.
Thank you, Lakshmi, for the talk. If
Thank
anybody
you.
is having any queries regarding protein engineering or small molecule, you can just put in the chat box. And also, like also have Gayatri, like who has given a talk about omics in like AI in omics workflows in the previous session. Pawan, if we have still time, like we have some exclusive like view for the exclusive view for the audience of the Nobel Research Sprint 2025. Like we haven't still released our product yet, but we are ready to show it to the audience of Nobel Research Sprint 2025. Like if you can permit some more time, like she'll just give a small overview and she'll be showing you the product.
Okay, so how much time it would take?
It will be around 10 to 15 minutes.
Okay, like we can do it for 10 minutes.
Yeah,
You
sure.
can go ahead happily. Yes, sure.
Yeah.
But
Thank you
let's
so
keep
much.
it for 10 minutes.
Yeah, definitely, sure. So we have Gayatri presenting. Actually, the tool has not been deployed yet for all the users. Like this is just the trial one that we are showing, so that the teams will get some insight and also they'll be able to frame their workflow around the tools that we have. Gayatri, please go ahead.
Hi everyone, am I audible?
Yes.
Yeah, okay. So you'll remember me from the last session. Today I'm here to talk about a very specific aspect of our Boltzmann suite. Like our experts have already talked about the different modules that are available, protein engineering, small molecule design. We are actually building and representing a team that's building another futuristic module of this called AI agents that you can see here. Okay, so far you've seen all these tools that are there in our platform, right? And what you can see are basically, it looks like a simple interface, but it is basically powered by very powerful, very fast and very accurate AI models, which are at your fingertips, even with the most complex of chemistry and biological problems, right? But then you would still need some expertise.
Hello? Hello, ma'am. Your voice is not audible now.
Uh, Pawan, am I audible?
Yes, yes, you're audible.
Okay, just give me a minute. There was technical issue. Like she's joining in a second. Just give me a few seconds.
Hello?
Hello?
Yeah. So I'll just so what I was saying is that you have these really powerful models that were built as a part of our Boltzmann discovery suite, and these are powered by like very fast, very accurate, very powerful deep learning and other AI models. But then you would still require some expertise to use them, to understand them, to interpret their results, and then integrate them into your problem, right? So what we are looking for is one step ahead. I want you all to imagine a room, a huge room, where all of these tools are present, right? Like an exhibition of sorts. What we are offering you in the next, in our next step is what the AI agent is. Imagine you have your own virtual lab assistant who isn't standing in this room, like an exhibition room where all these tools are present. All you have to do is tell to the assistant in normal words that I want to do this. And the assistant will be able to go select the tools that you want, plan the workflow for you, and then actually even, execute the tools and give you the results. The difference between this and ChatGPT is that GPT-like models, ChatGPT, Gemini, these models can take your queries and give you maybe an explanation from what's already there and maybe plan some solutions for you, which may not always be accurate, especially in the life sciences. But agents are powerful LLM powered models which are capable of taking your very simple queries, making it into a scientific question, and then dividing that question, planning your workflow, and actually doing the analysis for you. I'm going to quickly show you one such test that we conducted. So to save time, I've already run it for you. As you can see, this just looks like a chat GPT chat bot, right? All that they gave here is they uploaded a file and we said use the given file and just identify suitable binding area. So this is a protein file, the protein that Catherine showed you earlier. What the agent does is first it plans. So first it has taken your very simple query, use the given file and identify suitable binding areas. It has recognized what file it is. It has chosen a workflow. It chose a tool called binding site prediction from our suite, which is already existing in the Boltzmann suite. It selected a tool called fpocket. It is going to analyze protein structure to identify potential binding pockets. It has recognized the file that you have given that we have attached over here. The next step, it has generated the code. So because these are all in silico, that is, computer based, it has generated code to analyze your protein. It has taken the input, as you can see. It has made a workflow out of it, and it has successfully run it. In the execution report, you will see the workflow analysis. It has also performed the key metrics. If you were doing this on your own, you would have to input the protein, take the metrics, and then interpret the files. It has also given insights. Insights saying that the workflow was successfully completed and the pocket is located at the given 3D coordinates. The coordinates will be given to you. Other metrics that are there. And it also recommends the next steps, what you can do with the results obtained from this analysis. And similarly, give you download links. All you have to do is now just download this. I'll show you another analysis where we did like peptide property prediction. Here also what we just gave, like we talked to ChatGPT, we just gave a sequence and then we said, okay, run peptide generation flow on this. And we want peptide generation, prediction of generated peptide properties, and then structure prediction. So similarly, it has once again made a plan. It has understood the query, taken the sequence. It has done some preliminary calculations on the sequence, number of peptides, sequence length, classified these as inputs, and then it has generated the code responsible to run this execution. So as you can see, it's called a tool which is existing from in our platform already, and it has given all this information to it and run it. Once you run it, you can see execution and workflow steps and outputs. For each step, it will also show you the input and outputs. Similarly, for this workflow, you'll also be able to see what sequences were generated, output descriptions, everything that you would see in a standard result when you run this analysis by yourself. So what an AI agent essentially is a virtual assistant that currently enables you to plan a workflow from your normal queries. The queries, what we call natural language queries, how we talk to each other, and make a scientific workflow out of it. What they will also help you do soon enough, we are in the process of developing, is to discuss with you and come up with a plan. Imagine two scientists talking. So AI agents will be that for you very soon. So also, one other quick thing that I would like to add is last time we've talked about how omics can be automated. We're also building an omics agent. It's not currently integrated in the suite here, but I can show you a quick glimpse of what it looks like in the backend. Is this visible, my screen? Okay.
Yes, that is good.
Okay. I think we have problems today. Right. So what we're trying to do here is, like I told last time, right? Bulk RNA sequencing, what I'm highlighting on the screen, bulk RNA sequencing data, if you remember, is all transcriptomics data, gene expression, which is what helps you differentiate between normal and disease samples. Again, if I was doing this analysis, I would first download the data, then take the data, analyze it, first filter it, then normalize it because the data is very raw and very uneven, so to speak, when it comes from the experiments. Then we have to take what experiment was used to perform this, which is called metadata. And then we have to recognize what are the sample categories, how many normal samples are there, how many disease samples are there, and then perform what we call differential gene expression analysis. This also I discussed last time. It basically tells you what is the difference between a normal sample and a disease sample in terms of what genes are expressed. And then I would have to do what are the top differentially expressed genes, and then see if these genes have any mutations or not, because it's a cancer disease. So this is what I would have to do. But what I want to do is, why don't we just give this to an agent to run? This is an exclusive, back in the image of what we're trying to do. So what I'll do is, let's say I'm just going to run the file. This is what I've given as an example. And slowly, the agent, as you can see, is classifying the query. It has taken my input, It is classifying and then it is generating omics work. So what it basically does, it will give me the entire completed analysis of this. Since we're running low on time, I'll quickly show you the results which I've just run 2 minutes ago. So after you've given the query, what it has done is it has given me this folder. In this folder, what I have is it has taken my raw counts matrix. It has taken the entire data set that I have given it. Then it has performed differential gene expression. First, it has normalized it. So it has created another file for me in my own system. After doing this, it has performed differential gene expression analysis and also saved my file to the system. So now from this file, I know which gene, this is a list of genes, as you can see, gene, and I can tell which gene is expressed more in a disease sample compared to a normal sample. If it's negative, it's less compared to a normal sample. If it's positive, it's more. Similarly, it will also give me some plots. This is a volcano plot. It is also done another analysis for me, which I asked to see how many of these are likely to survive mutations. So we have these scores called PLI scores. You can see the graph, and it has gone and called that tool from the website, which is there on internet, and it has taken the data and has plotted the data for me. So essentially, to quickly wrap up, what agents can do is that first they take a plan from the user. They make a human-like plan out of it as if a scientist is making a plan, and then make tasks out of it like a to-do list, and then use their computational capabilities to perform each task and finally give you the result, save it into your own system. So basically, this is what we are aiming at Boltzmann as our next, venture is to not only give you an access of these extremely powerful tools for drug discovery and omics, but also we are trying to automate it for you so that you can use your abilities to come up with better problems and better solutions quickly, economically, and also you're using very powerful models. Thank you.
Thank you, Gayatri ma'am. That's
Quickly, 1 minute. I was also told that this will be available to you in your, I think, February sprint of your hackathon. So you could use this at that time. Thank you.
okay. Thank you so much, ma'am. That's such a great wrap up of the new tool. And also thank you, Dr. Catherine and Mr. Harsha, for the session on small molecule design and protein engineering. It's very insightful. So I think if participants have any doubt, they can put in the chat box. After this, I think we can wrap up the meet. Okay, I think no one has any question for now. But there is one question: when will this model be released? Okay, I think ma'am has answered this query. I'll just put you the link of the website of Nobel Research Sprint. So if you want to use this tool in the second round of our use this tool, you can register for an event, give your proposals with the given problem statement, and surely if you get selected for second round, then you can surely use this tool that we are providing. So thank you. I have provided the link below, so please use it to go through the website. Okay, then thank you everyone. Thank you for coming for this workshop. Okay, bye. Have a good day.
Thank you.